Can I just jump in and get started, Morgan? Okay, great. Welcome everybody. I am very excited to introduce our speaker today, Nicole Snyder, Dr. Nicole Snyder. She is a recent graduate of our M.A.S. in spatial analysis program here at Johns Hopkins. And I've got Nicole's bio in front of me, but it would take me probably 10 minutes to read through all of it. So since I know her more personally, since both Tim and I had the fortunate opportunity to teach her and train her, I can kind of speak to some personal aspects of her. You know, Nicole was a great student to have in class. She's one of those students that tests you with pushing the boundaries of what you're teaching, and really makes that worthwhile part on the faculty side of getting students through the program so much more enhanced. And we shouldn't be surprised by that from her, since I refer to her as Dr. Nicole Snyder, because she's a professor. She's a professor at Davidson's College in chemistry, right? So that's kind of a unique twist between public health and chemistry, and it's really speaks to the diversity of the students we have in our program. You know, I think she was our first professor that we had in our program. So just to kind of wrap this up, you know, so professor in chemistry, but I think for the last 5 years, she was kind of looking at different directions, and Nicole will tell you it was in response to her own midlife crisis. Not my words, her words. You know, some people buy fancy cars and go on fancy vacations. Nicole goes back to school, and so looking at here, there was an M.P.H degree, there was our master's in spatial analysis, and now she's still finishing up another GIS program. In addition to being professor in chemistry at Davidson College, she also serves as the public health epidemiologist for the North Carolina Wastewater Monitoring Network. So a lot of responsibilities, and we're excited to hear Nicole talk to us about spatial-temporal analysis of SARS-CoV-2 in North Carolina wastewater during the Omicron surge. So this is COVID-related stuff. So Nicole, I will give you the floor, and thank you for agreeing to come and talk to us today.
Yeah, absolutely. Thank you. It's my pleasure. I'm going to share my screen. I have some slides, so we'll get started there. And hopefully you can see my screen and the slides. Please let me know if you cannot.
They're up now.
Yep. Now they're up. Yeah.
Perfect. Let me see if I can get them into slideshow mode. Okay, so thank you very much for the kind introduction. It's really great to be back and to talk a little about the work that I did in the M.A.S. program. And as Frank said, the title of my talk is titled Spatio-temporal Analysis of SARS-CoV-2 in North Carolina Wastewater During the Omicron Surge, and the dates November 1st, 2021 through March 31st, 2022. And more, because I wanted to tell you a little bit about how I've kind of taken this forward to understand some of what we're seeing in wastewater, to start thinking about how we can build models and maybe even down the road do some forecasting. So to get started, I wanted to just begin with telling you a little bit about our program, because maybe, you know, everyone here may not be familiar with wastewater monitoring. So North Carolina has a wastewater monitoring network program that's actually part of the CDC's National Wastewater Surveillance System. And at this point, there are programs in all 50 states, but it didn't start that way. In fact, North Carolina was one of 8 initial sites that was funded by the CDC in 2021. They were funded at the state of North Carolina in 2020 to begin starting a wastewater surveillance program. And this was really all in response to the pandemic, right? So the COVID-19 pandemic. In addition to having support from the CDC, and the CDC does support the majority of the work that we do here at the state level, there are some sites in North Carolina that are being supported through a federal wastewater testing contract. Verily is the company that does that work, and they have about 300 sites across the United States. They have 3 in North Carolina. And then we also have a couple of sites that participate in the wastewater scan initiative. The map that I'm showing you is basically providing you with the different sites where wastewater monitoring occurs across the United States. And one of the things that's really exciting for us is that North Carolina is one of the first to pop up there because of our engagement early on. And another part that's really exciting is last year in August, the CDC named us a Center of Excellence. And so we are one of 6 centers of excellence across the United States. We are focusing in on the Southeast region, so Kentucky, Tennessee, Mississippi, Alabama, Georgia, South Carolina, and Florida. And the idea behind these centers of excellence was that these COEs would serve as regional leaders, and they would really help to kind of support the surrounding states as they develop and expand out on their wastewater program. And the other thing that I want to note here is that this work isn't always done just with us in Department of Health and Human Services, but we do have an academic partner, the University of North Carolina, and also we do have a wastewater treatment plant partner, OWASA, which is in Orange County, and that county is where UNC Chapel Hill is located. Okay, so why wastewater, right? Why are we interested in this? And a big part of this is because wastewater can serve as a really important community-based monitoring tool. We've known for a while that it can serve as an early warning tool in some cases to try to detect the spread of disease. And COVID-19 was a great model to do wastewater surveillance with. So some really I think, positive aspects of using wastewater monitoring. First, it captures information on all types of COVID-19 infections. So you'll have people who are symptomatic and they may seek care. There'll be people who are asymptomatic and they don't seek care. But as long as they're using the toilet, and as long as that toilet, then the waste from that toilet makes its way to a wastewater treatment plant, in principle, we can monitor it, right? And it turns out that it's widely applicable. Across the United States, about 80% of US households are in fact served by municipal wastewater collection systems, right? Now, it's a little different in North Carolina. We're about 50%. We have a large percentage of our population that is rural and that are on septic tanks, but we can still capture about 50% of the population. The early detection, I mentioned, the early warning tool. In some cases, wastewater can show changes in trends from, 4 to 6 days before those changes are seen in clinical cases. And we're focusing on SARS-CoV-2 for this presentation, but I'll tell you that that's also the case in some cases for other types of pathogens like flu, for example, and even RSV. Wastewater is a great way to look at changes in the variants of different pathogens. It has been very important for COVID-19. So you can get a community-based sample that kind of gives you a sense of which variants are spreading within the community, or at least which ones are most prominent. And then one thing that we like is it's independent of medical systems. We know that not everyone has access to care, right? And that some people have more access than others. And so again, as long as people are using that toilet, right, even if they're sick and not seeking care, we can capture that infection in the wastewater system. And then last but not least, I think it's really important just to consider that while wastewater surveillance has been around for a very long time. People looked for polio in wastewater, 50 years back, but it's really gained prominence with SARS-CoV-2 and with the pandemic, but we're now tracking a number of other pathogens, flu, RSV, M.P.H. We're looking at flu variants, and there's a lot of other conversations that are happening around measles, right? And so that may be a direction that we ultimately go in the future as well. So how does it work? I like to call it from bathroom to benchtop. The physician scientists always like the benchtop to bedside, but ours is bathroom to benchtop. And what we can see over here, and this is just an infographic that was put together by the Department of Health and Human Services, right? Is you have the community contributes waste. That waste then has a residence time, travels to the wastewater treatment plant. And our wastewater treatment plant folks are the ones that actually collect those wastewater samples and ship them to us. And I have to say, they have been doing this really for almost 5 to 6 years, depending upon when they started. And in our case, in North Carolina, they're doing this, pro bono. They're not being paid for this, right? So it's really a volunteer, and we're really on a volunteer basis, and we're really grateful for that because at any one point in time, they could stop. They could decide that they don't want to do this anymore. But they've continued even post-pandemic and have been really on board with some of the other types of pathogens that we've wanted to sample. Those samples are transferred to either the state lab for public health or one of our academic partners, and then they analyze the wastewater that causes COVID-19 or other pandemics. Again, this was put together during COVID-19. This is where I come in. I'm the epidemiologist for the wastewater surveillance team, and so I do some data processing and use that data to populate a lot of our publicly facing dashboards. But I also have to interface with the CDC. I submit that data through the CDC portal, which is called One CDCP, and then the CDC analyzes that data, analyzes trends, and then ultimately sends information back to us. And we use some of their reporting as well. So it's kind of a dual reporting process where we do some in-house, and then we actually use some of the reporting that comes from the CDC. And then, of course, I mentioned, the results being posted on the dashboard. This is just one example of the Cherokee wastewater treatment plant. This is in the Eastern Band of Cherokee Indians, and they are one of our partners. But it gives you a sense of what these wastewater treatment plants look like, and they can be relatively small, like the Cherokee plant, to really large, for example, like Charlotte, Mecklenburg, or Raleigh. I just put these pictures here so you can see what this process looks like, right? These are examples that were taken by some of our partners. We send out kits each week. They collect the samples, they put the samples back into the kits, and then they send them to whatever lab is being tested. And so this just looks like this is just a good example to show kind of what that sampling looks like. And then this is the process, and this becomes really important because it helps you to understand some of the ways that we're looking at this data. So we collect the data. One of the first things that happens when it arrives at the lab is it's heat pasteurized, and there's some serious reasons for that, right? We're looking at pathogens in wastewater, and there's all kinds of things other than what we're maybe looking for. And so this just protects the workers in the lab. They need to then add some controls, filter and concentrate, and then they're extracting the nucleic acids that are specific to what other pathogen they're looking for. They then use RT-PCR, right? And that's how we get the concentrations. And I want to say that when we get those concentrations, we actually do some normalization. We normalize it by two things. First, we normalize it by the flow rate, right? That's very, very important because we are in North Carolina. There are times when we have really heavy rain. And so you've got a lot of, storm water and runoff that's coming into those wastewater treatment plants that can dilute the sample. We also have hurricanes and other weather events, and so it's important to normalize by flow. And we also normalize by population because, depending upon the wastewater treatment plant where we are sampling, right, that might represent a very small population. Our smallest are around 3,000 to a very large population, which can be upwards of close to 100,000. So it's really important to try to have a method for normalization. And so we report those out as viral particles per person per liter. Okay, so we are routinely monitoring at 35 different sites across the state of North Carolina. We're monitoring for 4 respiratory pathogens. I'll be talking about SARS-CoV-2 today, but as I mentioned, we are looking at influenza A and B. We've really been looking at influenza A and particularly subtyping for H5 as we've had some H5 detections in North Carolina, and then also for RSV. I'm not showing M.P.H here, it's not a respiratory pathogen, but we do regular monitoring of M.P.H at several of our sites across the state. I just thought I'd give you some statistics just so you understand the scope of what wastewater can do in terms of representation across the state. This is kind of what we call our wastewater wrapped, kind of like a Spotify wrap for 2024. I mentioned that we're looking routinely at 5 pathogens at 35 sites already, but we've collected almost 4,000 samples across 2024. When you multiply that through by measuring for 4 to 5 pathogens and then doing all those sampling all those sampling measurements in triplicate. It's a lot of samples that the 4 people that work at the State Lab for Public Health manage. So it's quite a bit. And we do have a partner again at UNC that does some of that work, but it's a lot of work. It represents about 30% of the population in North Carolina, so close to 3 million people, over 36 of the 100 counties, 42 emergency departments and 329 long-term care facilities fall within the sewer sheds. Those are the regions that essentially are surrounding these wastewater treatment plants that then contribute to them, as well as 797 public schools. So we're reaching a large swath of the population. And remember, 50% of our population in North Carolina is about 10 million are in septic systems. So even if we maxed out, we'd only be getting about 5 million people across the state. And I also think it's really important when you're doing analyses like this to look at how representative the data is. And we've done some really nice, we published a paper in PLOS1 in 2024 with work with Mathematica that, use some spatial work to look at whether or not our monitored populations really resemble our statewide populations. And what we found is that they do, generally both at the countywide and at the statewide level. But we also noted through that we are oversampling, persons who may be more vulnerable, right? So, if you look at, for example, our social vulnerability indices, we're a little bit lower there with the monitored versus the mean. But if you look at underrepresented populations like African Americans and Hispanics, we're tending to oversample there, as well as from people who fall below the federal poverty line. So it's a really, really nice way to show that what we're doing is really providing information that can be representative of the people that live in our state, even though we may be only sampling at a few select sites across the state. Okay, the last thing that I wanted to share, because I think sometimes we can get so caught up in the academic elements of the work that we do, is that we forget how the people that we work for and the people that we work with actually use this data. And so I put these, short testimonials here, one from an anonymous dashboard user, a community member, and then one from Buncombe County Health Director. And these are shared with permission. And the first one says, as an immunocompromised woman, current wastewater surveillance data is crucial for me. The North Carolina Wastewater Monitoring Dashboard has been a trusted bright light in North Carolina public health the past several years. It's a gem that is not only available in every state, and I do not take it for granted. My family uses NC's wastewater surveillance data to choose when to schedule non-urgent doctors and dentist appointments and to decide whether we attend group activities or not. Knowing the level of community infections is the difference between going blind into a COVID surge or being armed with the correct and current information to increase measures to stay safe and healthy. And I think this is more important, especially once the pandemic, the emergency that ended in 2023, because we don't have a lot of case data right now, right? So wastewater becomes one of those only ways that community members can really get a sense of what's going on. And then at the county level, the Buncombe County Health Director stated, wastewater surveillance is a valuable tool for local health departments to track illnesses like flu and COVID in our communities. We use the information to inform health messaging and encourage public health action like vaccination or notifications to local healthcare providers. And so, again, you can see that use, at the local health department level. So again, I put this here more just to say that, it's really important, the academic work that we do with this data, but it's also super important to the communities that we're serving, right? And we are funded by the taxpayer. So it's really important to make sure that value is there. Okay, so now I want to turn to the work that I did. We talked a little bit about the title, but I wanted to kind of go in with the question that I was hoping to answer when I started my work. And that question was to really examine wastewater and case data. And first, thinking about how well they correlated over space and time and how we could use this data to inform public health. And so this is the work that I did as my integrative activity at the end of my degree for the M.A.S. in spatial analysis for public health. And so I wanted to start by looking at what the goals of my project were, and I'm going to walk you through two of those goals and the results that I had, from the work that I did. So the first goal that I had when I started my integrative activity was I wanted to compare the onset of SARS-CoV-2, particularly onset of the surge in both clinical and wastewater samples. And I wanted to do this across 25 different sewer sheds. And I had a reason for this, right? When I'm doing work and that work is for, our wastewater monitoring program, there's always the what is the public health benefit, right? Why are you doing this work, right? What can it help us to do as public health professionals? And so for me, I was hoping to look at lead times, right? There's a lot of evidence out there that in certain cases you can detect SARS-CoV-2 in wastewater well before you can actually see it in the clinical cases. And so I wanted to kind of test that hypothesis, because if we do really have a lead time, that can play an important role in operationalizing public health measures. So if you know that, for example, you generally start to see increases in SARS-CoV-2, for example, in a certain region of the state, and that happens, over over time with a number of different surges, right? We're looking at one here. Then that can help us to maybe use that site as a sentinel site to say, oh, I think we're going to see a surge, right? Let's start rolling out some public health measures. So this was a really important question. The second question that I was interested in is kind of tied to that first goal. How does it actually move through the state of North Carolina over space and time? Do we see that there's regions that sort of light up early, right? Again, going back to that sentinel surveillance idea. And so that's part of the reason why. And these goals are kind of intertwined, right? But they were important goals for us to think about how we're using the sites that we have across the state to serve the population the best. And I will say, many of you know that there are, significant challenges right now in public health funding. We've been lucky to maintain the majority of our sites, but we also know that at some point in time, we're probably going to have to scale back, right? It's going to become, a point where we won't be able to necessarily sample at all 35 of the sites that we're currently sampling at. And so, understanding which sites generally tend to correlate best with what we see in the clinic, and then also, which sites tend to give us those early warning signals are definitely ones we want to keep, the ones that we want to continue to invest in. And then the third goal that I had was to evaluate whether any relationship existed between where the cases are, so where we know the cases are within a perhaps sewer shed, and then, whether that had any relationship to what we would see with the wastewater signal in terms of its distance to the wastewater treatment plant. I'm actually not going to talk about this part today. I'm happy to tell you a little bit. One of the things that we've been finding, that I found both within my integrative activity, but, since as we've really worked on this problem is that, the locations of the houses that contribute, the sites that contribute even within the sewer shed really aren't that far enough away from the wastewater treatment plants to see any significant degradation of the signal. We were worried that, perhaps people who are contributing to that signal that live, 5 to 10 miles from the wastewater treatment plant or even further, that, as that is deposited and makes its way to the wastewater treatment plant, that there would be some degradation of the signal just from the breakdown of the RNA that we're working to detect. But we haven't really seen any evidence of that. So I'm not going to talk about that today, but I am going to talk about the first two goals. Okay, so the first slide that I wanted to use here to just kind of talk about the work that I did is some epi curves, right? So in this case, one of the goals of this initially was to look at the Omicron surge at 47 sites. I noted that I took that back to 25, and there's a reason for that. I'll tell you that in a moment. But I did use the data in total to get a sense of, what these epi curves look like. And so what we're looking at here is November 1st through March 31st. So November 1st, 2021 through March 31st, 2022. And what we can see is you have these are SARS-CoV-2 cases here in the top graph in green, right? And then this is the SARS-CoV-2 average particles per person in wastewater, right? That's that measurement that we use for wastewater over that same time period. And in this case, right, without going into the details and too much detail, what we can see is that there's this flat, kind of flat here, and then all of a sudden, right, as we start to get into late November, right, and into December and January, we see that the number of cases increases. And if you look at the wastewater signal, right, we also see, generally the same trend, right, that these that the signal overall increases. And just one thing to keep in mind, right, these dates, November 2020 to March 31st, 2022, right, we were still getting we were getting case data every day, right? That isn't the case now. So it's harder to do a study like this with more recent data, though we do have some syndromic surveillance mechanisms for getting, at least some information on who's visiting the emergency department. But nevertheless, I think it's just important to note that, the reason why I even chose this time frame was partly because we did have the case data, right? And so I could use that as more of the gold standard, right? And then look at wastewater, as kind of the measurement, right, in comparison to that gold standard. The other thing that I want to note about this, and just something to keep in mind as I walk you through this study, is that, towards the end of 2021, 2022, testing, home testing became more frequent, right? People were able to get access to home tests. And so in many cases, they weren't reporting those home tests, or maybe they would test themselves and just isolate. They wouldn't seek medical care if they weren't feeling particularly bad. And so, we also have to understand that, we were probably seeing some reduced testing, towards the end of even the Omicron surge, just based on the availability of those home testing kits. So there's some caveats here, but, for the most part, we do have some good case data for this time frame. And of course, we have our wastewater data. Now, I mentioned that, even though I'm showing the data for I think I did call these down to the 25 sites. We had originally been examining, data at 47 sites. I chose to use 25 sites, sorry, partly because I wanted consistent sampling. And what I meant by that is that at the time we had 3 different laboratories that were doing the measurements. And unfortunately, not all those laboratories were in sync. And so while you could compare perhaps, average particles per person in wastewater across the sites that were being sampled and then measured within a specific lab, it's a little bit harder to compare them across labs. So I chose to work with the sites that were being sampled by the State Lab for Public Health. The other thing that I want to mention before we get started here is on this map that I'm showing, there are these yellow dots, right? These are the wastewater treatment plant centroids. And then I'm also showing the sewer sheds, right? So this is the geographic region where the people that generally reside in that region, maybe not necessarily everyone, right? But a large percentage of the people that are in that particular geographic region are contributing to the signal at that wastewater treatment plant. Okay, so that gives you a good sense that these are larger geographic regions in some cases, right? You had a big one here, and then you have these little small ones that almost look like points themselves. So just something else to keep in mind. Okay, so data. The data that I used from the analysis came from the North Carolina Department of Health and Human Services, and I used this data with permission. I had the raw and processed wastewater data. The variables are included here, but in general, the ones that were most important to me were the wastewater treatment site locations. I needed the sample ID for joining because I had to do some joining work. I had a site identifier that was also important, in case I didn't want to, release names, things like this. Population served within that sewer shed was important, even though I already had the normalized wastewater signals. I still kept that population there just, to hold on to that. And then I also had sample collect date, which was really important for this study, especially for the temporal element, flow rate, and then, some additional information. I actually did have the 95% confidence intervals for the data. I didn't end up using them. I treated the data as hard data for this study, but that's something that I've always wanted to go back and do is kind of, look at that data with that error. The case data, as I mentioned, were also obtained from DHHS. I had the home address, I had the type of detect, whether it was an individual, whether it was part of an outbreak. I had whether or not the patient died. I had information that was also identifying, and didn't end up using all of this data in the study. It wasn't crucial for the work that I was doing, but I did need information on the home address because I had to geocode those cases to the different sewer sheds so that I could get cases per sewer shed as a comparable for this particular study. And then I also obtained shapefiles. I had the shapefile for the sewer shed boundaries and for the wastewater treatment plant locations, the points. And then I also took a shapefile for the state of North Carolina just for visualization purposes, but I didn't actually use that. Okay, so that gives you a sense of the data that I had available to me. I was very privileged to be able to have this kind of data because it essentially gave me a complete picture for the study period that I was interested in pursuing for my integrative activity. Okay, I did want to, before we hop into some of the results, note that I did have to do some data aggregation. So for wastewater data, we sample at each wastewater treatment plant location twice per week, and each sample is measured in triplicate. So what I did was I essentially averaged that data over a week, right? So I took those two samples and averaged the average concentrations. For the case data, as I mentioned, I had case data for the entire state. So what I had to do was I had to figure out which cases were located in each sewer shed. I've shown an example of how I've done that. And of course, to not give away any identifying information, these are kind of zoomed out, but you can get the picture. I essentially use these sewer shed boundaries in order to capture those cases over time. And so this is just kind of interesting where you can see kind of the onset of the surge, right? And then we're kind of heading into it, right? It gets more concentrated and so on and so forth. But I did this for each and every week, right? So I kind of aggregated these by week. So hopefully that just gives you a sense of how I aggregated the data, but it made sense for me to do it by week. And there's the reason for that is twofold. We get case data at that time every day, right? So the case data was reported each and every day. Unfortunately, though, with the wastewater treatment plants, it just depends on which day they choose to sample. And so if you don't aggregate it by week, it's going to be a little bit trickier to do this analysis because you have some that are sampling on Monday, some that are sampling on Tuesday, some that are sampling on Wednesday, and so on and so forth. So just having a weekly aggregate just made more sense for the type of study that I was working on. Okay, so research methods, what I did, and then we'll look at the results here. So I actually used a program called BME GUI, and this is a program that I selected because it allowed me to use this Bayesian maximum entropy, and that was selected to try to minimize the bias, like missing values and gaps in observations that are kind of endemic with wastewater data. And this was a program that I learned to use through some work that I had done in my M.P.H. at the University of North Carolina at Chapel Hill under the direction of Mark Sayre. And I was really grateful that Tim and Frank let me continue to use this for this particular study because I thought that it would be a really interesting program to be able to use to study wastewater. And so this is the program that I ultimately used for my work. And then I generated all the Krieg maps that you'll see. That was actually done in ArcPro. I did use log normalized data because the data was heavily positively skewed. And so that's an important thing to keep in mind as well. The first thing that I did was model the mean trend, and then I removed this, and I did this to be able to better estimate some of the more residual small scale variance that would be due to the finer spatio-temporal resolution. So I'll show you what that mean trend looks like and just kind of, really kind of flattened it, to be honest. And then I generated a covariance model, and the purpose of this was really to quantify the space-time variability. And I used that model to generate the maps of the Krieg estimates and variance over the study period. I'm not going to show you all of them because you could, in theory, do one for each day, but I'm going to show you kind of beginning as we enter height of the surge and then on the downside, so you get a sense of what I was able to do with this. And then, of course, generating those maps, and that was done in ArcPro. Okay, so first, the very first thing that you should do with any data is to do an exploratory analysis, and that was, important here as well. What we're looking at here first is the case data, and you can see pretty clearly from this top histogram here that in this case where we've got COVID-19 cases on the x-axis and frequency here on the y-axis, that the data is heavily right skewed or positively skewed. And so it was really important for me to do a log transformation in order to get a relatively normal distribution. And that's what you're seeing here on the histogram at the bottom, right, where you can see that the data is more normally distributed. I did look at the mean 9.56 cases per 10,000 persons at the sewer shed level and the standard deviation as well. And I did this for the wastewater, right? And perhaps unsurprisingly, it was also heavily right skewed. And so I did a log transformation. So this is ultimately what the data looked like. I used those log transformed values to do the remainder of the analysis. Okay, I did want to model the mean trend. I did this first for the case data. And so what you're looking at here on the right-hand side is the spatial and then the temporal data. And one of the things that, I really wanted to do was to think about flattening that mean trend so that I could focus on local variations. And so to do that, I used a search radius that was 2.1 degrees for the spatial mean and 60 days for the temporal mean. And then for the smoothing range, I used 0.7 degrees and 30 days for the temporal. And I did this also for the wastewater. And I'm going to show you the next slide because it's a comparison of the two, right? And so you can see that those the spatial mean trends And the temporal mean trends are shown for the case data on the top and for the wastewater on the bottom, right? We can see some relatively significant differences in some places, right, for the for the spatial mean trend. For the temporal mean trend, right, we have it's kind of remarkable how similar they are, but then maybe not. I mean, we're modeling an infectious disease, and we're seeing a pretty close relationship between what we're seeing in case and wastewater data. We knew that from the epi curve, right? But one thing that we tried to do was flatten this curve a little bit. So more than one ways to flatten a curve. And in this case, we did this when modeling our mean trend. So we flattened that mean trend again to kind of focus on local variations. Okay, this is a busy slide, right? But the next thing that we wanted to do was develop the model, right? So there's a spatial component and there's a temporal component. And I put the equation here for the spatial component. I put the equation here for the temporal component. I'm happy to talk more about each of these. I do want to show you the variograms for these, but bringing these two together gave us what I'm considering the full model. And I want to note that this is a 3-component model. I actually built in some measurement error. So just kind of doing a back of the envelope calculation of what we knew about the wastewater data and the amount of error in sampling. And then also what we knew about, perhaps cases that we weren't seeing because of the testing, particularly towards the end of that surge, because people were using those at-home testing kits that weren't necessarily being reported. We worked in the error, about a 10% error. So that first component is really meant to resemble the measurement error. And then we have this short-range spatial variance and then the long-range spatial variance in these other components for the model. So this took some time to develop. And I had, I have to say, when I first started, I had a really strong background from my work in the M.A.S. program, but I'd never really, thought about working with a 3-component full model. And so, it took a little bit of playing around and thinking and a lot of background work to try to figure this out. But yeah, ultimately, this is what I chose to work with. And this is what it looks like. So this is for the case data first, right? So the top is the spatial, the bottom is the temporal. So spatial component and temporal component. These are the variograms. And this is the model for the clinical data, right? For the case data. Again, just to remind you, this includes this kind of small kind of work the nugget into the model here, right? To account for the measurement error that we estimated based on, having those 95% confidence intervals from the data. Again, I treated this data as hard. I'd love to go back and really consider the error since we have it, right? We have those confidence intervals, but this was my first pass at this and I was kind of learning along the way. So it's really just easier for me to do it this way and to use up more of a simple kriging method than to do something like a Poisson, which again, I'd like to do someday, but that's not where I was at this point. And it's just, this gave us a good first pass to see what we were looking at. What I learned from this model is that it seems like the long range term really contributes most to the variation. And these fits, I mean, if you're looking at this, especially down here, this is rough, right? So this is kind of the best. Again, it took a long time in trying to play around and see what I could do to get the best fit that I could for this data. Knowing that you have this significant dip here. It doesn't behave like the data we got in class. It's real data, but did my best, right? And this was the model that I ultimately developed for the clinical. It shouldn't surprise you that the wastewater data would look similar, and that's what we're seeing here. So again, I included that term to help with the error that we know is associated with wastewater data. And then, looked at the model, and it looks like in this case, more of our short-term and long-range terms do contribute significantly to the overall variation. You can see kind of a side-by-side here. There's differences, of course, right? But you do tend to see some similarities in the variograms here for the spatial and temporal components of this data. So those are the models that I developed. They're not perfect, right? There's a lot of work that's still to be done. But as a first pass, it would at least give me some insight into what we were seeing with the case data and with the wastewater data. Okay, so I'm going to turn my attention to the Krieg maps that I generated using these models. And I put these yellow lines here to indicate, where I chose to do some of my mapping, right? So I wanted to map a date that's really kind of representing a week, if you will, that was kind of pre-onset, right? So like maybe just before, at least from these epi curves, looked like it was right before the onset of the surge. And then right at that beginning of the surge, kind of in the middle of the surge, right? And then towards as the surge was kind of decreasing, right? And then in March before we went into the summer surge, because we had another surge. So one thing that I want to point out here is that I chose, I tried to be very careful when I chose. We had some irregular sampling around the holidays in some cases, and so I really wanted to pick something that was at the height, but maybe outside of that New Year's. That year we didn't get quite as many samples. You can kind of see around New Year's, right? And then sometimes around Christmas, you get a reduction in sampling because people are on holiday. So that factored into my decisions as well. So those 5 dates are the dates that you're going to see the maps for. And this is what they look like. So these are the Krieg maps that I generated. These are the BME means estimates. And this is SARS-CoV-2. And then, of course, the estimation of variants. And this is for the cases. And I should put the cases and the concentrations here, so I apologize. But one thing that I want to say is that our wastewater is always going to be here over on the left side, right? Our cases, our clinical data is going to be on the right side for these maps. The top maps are representing either the log copies per person, right, or the log cases per 10,000 persons. And then these are the estimations of variance. So these aren't going to change because they're consistent for the model, right? But I put them there just so that you understand that, one of the challenges with this data, as you notice from the maps, we have these sites that represent sewer sheds. For this study, I kept them as sites, right? I used the centroids. And so we get these little islands, right, where we have less variation, and then we have the areas in between where there's significant variation. And there's not much that I could do about that given the number of sites that we're sampling across the state. We see the same thing for clinical data, although there appears to be less variance. And so, again, those maps will stay the same. But the takeaway from these, so starting with November 12th, 2020, this should say, 2021. There's a typo there. In this case, what we're seeing here is these red spots, right? This is in the Charlotte region. This is kind of up in the Winston-Salem region, right? Maybe a little bit out here on the western part of the state, right? And then up here towards Virginia, right? That's what we're seeing in wastewater. With the cases, we're kind of seeing red spots out here in Buncombe, kind of near Asheville region, and then in Raleigh. So we're not really seeing, the same hotspots between these two maps. And at least from the outset, it looks like you're seeing a lot more signal here in the wastewater than what you're actually seeing, in the cases. If we start to look and get a little bit closer, right, because there's a little bit more variation, frankly, out there in early November. If we start to look and get, somewhat closer to, for example, the actual surge, right? So now we're looking at December 23rd. You can see it's pretty much red everywhere, right? So in the western part of the state, towards the central part of the state, looks a little cooler out here in the western part of the state for the case data. But, we were, headed right into the surge, and you're starting to see significant signals, across the state. What becomes interesting, right? Here's the height of the surge, right? With wastewater, you're still seeing really heavy signals throughout the state, right? So you see that signal persisting in wastewater, but you're starting to see those signals diminish in the case data, right? We're January 14th, which is the peak. So you're seeing a lot of cases that are being reported, but you're not necessarily, you're seeing far more, at least, signal here in the wastewater. And then as we go on as we start to head down on the other side of the surge, right, we see those signals still persisting in wastewater, whereas they're largely clearing out when we start looking at the case data. And then finally, by the time we get to March, right, you start to see a little bit more building back here in the West. We had a surge in the summer, so we started to see cases slowly trickle up. But for the most part, right, everything's pretty blue, right? Whereas over here in the wastewater data, we're starting to see some more hotspots, again, here in the Charlotte region, and this time up in the Raleigh region. So, in general, when I looked at this data, which you could see is slowly, it seemed like, for the most part, we started to see signal in the western part of the state in the Charlotte region, sometimes up in the Winston-Salem region, and then it would move more towards the Raleigh region. And I'll show you some more recent data that we've been looking at with another surge where we see that pattern as well. So it seems like for our state, it kind of moves from west to east. I did want to look at these maps overlaid with the cases, right? So actually use the cases that were in each of these sewer sheds. And again, these are kind of zoomed in, so you can't see the exact point locations, but trust me that they are there, right? And these are the dates: November 12th, January 14th, and March 14th. So beginning kind of at the height and at the end, right? And so in this case, we're looking at wastewater data on the left-hand side and then the case data on the right-hand side. And it's really interesting because in November, right, we start to see larger amounts of cases where we're actually seeing heavy wastewater signals, right? Except for here, right? That seems to be better predicted by the model that we're using for the clinical data, but then it should match for the clinical data. It's the model that's based off of the clinical data points. But it was really interesting to me to see that, sites where we were seeing significant numbers of cases within those sewer sheds were lining up much better with the wastewater data than they were with the case data, right? Now, again, when you're looking at the Raleigh-Durham region, right, the model here for the case data seems a bit better. At the height of the surge, it's tough because everything is everywhere in the wastewater model, right? There's a lot of heat on this map, and there's a lot of cases, right? But again, we can see some significant differences, right? Where, for example, in the Winston-Salem region, it's a little less hot, where it's pretty hot here with the model that we've built for the clinical data. But then you have this cooler region where we see a significant number of cases in the Raleigh-Durham region, right? And then it's more hot here on the wastewater map. So kind of, some interesting points to make there. And then as we sort of came down from that surge, right? We still see like some significant heat here in the Raleigh-Durham region, right, on the wastewater map, but not really so much for the model that we developed for the cases. And so, what does all of this mean? There's a couple things that it can mean, right? And we'll talk about that here in just a quick moment. So here it is, right? It's complicated, right? In some cases, some sites, if you start to break it down by a site by site, what you're going to see is that in some cases, right, some sites are predicted better by the model than others, right? So I'm giving an example here where we're looking at the Cary sites. And this is just an example where we can compare the wastewater model over time to the clinical model over time. And we can see that they both do a pretty good job at early onset, right? But as we start to move into that surge, right, the wastewater model does a much better job at predicting what we see with the cases than the actual case model itself, which is interesting to me, right? And that's not all across the board, right? And there may be reasons for that, right? Because, wastewater, you tend to see a more persistent signal. People may be, they may be infected, but they're not necessarily going to the doctor. That home testing, right, more towards the end could be affecting this as well. So there's a lot of factors here that could be affecting why this model is kind of falling apart here at the end, where the wastewater model tends to better represent what's happening in the population. But at the end of the day, right, I think the take-home message is that the wastewater model does at least a pretty good job in this particular region. But if you go region by region, that's not always necessarily the case, right? So that was interesting to me as well, just to start thinking a little bit more about what happens when you're looking at the individual sewer shed level. So I'm getting close to the end here. I know I'm close to my time. I just have a couple more slides to show you. We have been comparing a lot of our wastewater data to clinical data, and we've been trying to look at that early onset. That was something that we were interested in and kind of saw with the model that I just showed you, but it's tricky, right? In 2023, this was an example where we really saw wastewater started to increase well in advance of what we were seeing in emergency department hospitalizations. And in this case, we saw wastewater increasing almost 3 weeks, right? Before we saw before we started seeing those cases in the clinic, right? And then about 7 weeks before it ultimately peaked. And so we wanted to look at this as well. And I just have one slide to show you, but I thought this was really nice because I used the same model that I developed. And we looked at June, right, which was the early onset, September, and then October. And this was kind of cool because we actually saw this pretty clear, right? So what you're seeing over here is the model. Here's the variance from the model, right? And this is just wastewater. There's no clinical data in this one, but we see, at the onset, this really large signal here. It's pretty hot right here in the Charlotte region, right? And then as we get towards September, it's still pretty hot in the Charlotte region, but moving over towards that Raleigh-Durham region, right? And then as we get more towards October, right, we start to see that signal dissipating here in the Charlotte region, but we're still pretty hot over here in the Raleigh-Durham region. So you do kind of see that onset, right, and then that movement, from west to east across the state. So that was interesting to see where we could see it, right? We knew it was there as we were monitoring the clinical data and the wastewater data, but also really nice to model it to be able to show that movement. It's been really interesting. I'll say this as an aside because I've been doing a lot of correlation analyses as well. The wastewater data is aligning much better with our syndromic surveillance data than it did with the case data. And I don't know why that is, but anyway, it's interesting. Okay, so summary. Signals appear to move, at least, somewhat, right, from west to east across North Carolina. Wastewater signals do generally tend to appear first, right, but there is some significant variation by site. SARS-CoV-2 signals do seem to persist longer in wastewater, and that may be because people are continuing to shed virus long after they're, no longer infected. We know that, but some people shed for even longer than others. Visually, the case data sometimes better. Visually, the case data align better with the space-time model for wastewater in most cases, not all. But that was something that was really interesting. And I do think that's a factor of testing, especially towards the end of that surge. I think one of the things, the conclusions that I could draw from this is that the spatio-temporal analysis could be used to examine wastewater and clinical case trends. And I think it's a really great way to try to use this data. It is really tricky. And I also learned that, through my work in the M.A.S, it's a short program, right? And I learned a lot in that program, but I still have so much to learn, right? And that's the one thing that I've learned from all of this work is I know, I know a lot, but I know less than I would like to know. So I'm always continuing to learn. Limitations, we only have so many sites and so much data that we could use for the study. So that's always a limitation. We got these kind of creaking islands all over the map. Wastewater data is messy. These are complex samples. They're collected as 24-hour composites. They're filtered. How they're measured even on the filter can change sometimes the outcome of what's reported. I treated the data as hard data. I didn't take into account the error that was associated with the data, and then I kind of built my own nugget into the model to account for that measurement error. So, going back and actually using the real error that's associated with all of those samples would be a better way to do this. It's a little bit more challenging and a little bit more tough for the time frame for the integrative activity, but it's something that I would really like to go back and do at some point. And then finally, the model hasn't been cross-validated for accuracy and reliability. And so this is something that would also need to be done. But, again, time. So as a last slide, I'll leave you with where we're going. We have a lot of data. We're also trying to start using syndromic surveillance data since we no longer have case data. Kind of alluded to the fact that we're getting some better data or some better correlations with that data. And then we're also trying to use some wastewater sequencing data to do some analyses. And then last but not least, we're still using GIS data to do even just, some of our more simpler work. Like as we try to think about, we had the opportunity to expand to other wastewater treatment plant sites. These are the locations on the right-hand side here, all the wastewater treatment plant sites across the state. We had a chance with the CDC to expand to 10 additional sites that they would fund through their contract program, Verily. And so we did, we used GIS to do an analysis to look at things like where are emergency departments? What are the drive times to those emergency departments from these different, sites? And how could we look at, that combined with things that we know like social vulnerability that are important to be able to select those sites. So we're doing a lot of that work using GIS to select new sites just to make sure that we have better representation of our population. Okay, so with that, I'd like to acknowledge Frank and Tim for supporting me throughout the program. I really benefited from their mentorship, and like I said, I learned so much through the program. Also, Mark Sarah at UNC, who was very instrumental in supporting me back when I was working through my M.P.H. And between Frank, Tim, and Mark, I decided to really go the GIS route, and I'm still learning. I'll continually be learning as new tools are developed, but I feel like I got a great start. And so with that, I'd like to thank you for coming, and I'm happy to take any questions.
Thank you, Nicole. I think that was great. Um, and I think there are some questions in the Q&A, and I was just kind of scared by one thing you said is that there's still so much to learn. So I guess there's more degrees and more programs for you to involve yourself in the future.
Oh,
So
come on, Frank. We're always learning.
yes, yes, we are.
Yeah, let me see if there's let me look in the chat here if I can see. Oh, that just says
Here's
Q&A.
the Q&A.
Okay, let me see. Q&A. Okay, got it. All right. So William writes, can see the connection between wastewater analysis and chemistry. Many thanks for a great presentation. I need to attend another meeting. Good luck in the future. So thanks, William. Sorry you're no longer here, but I appreciate that. And then Charlotte, did I understand correctly that wastewater data are urban dwellers, workers, but case data are urban and rural dwellers, workers? Can you split out the case data to urban dwellers, workers? Yeah, so this is a great question. So the wastewater data represents pretty much anybody that's living within a given sewer shed, right? And so, but that also includes people who may be traveling into that sewer shed, right? So this is a little bit tricky because sometimes we can get a signal, and I'll use M.P.H as an example. We can get a signal for M.P.H, for example, in Mecklenburg, right? In one of our Charlotte wastewater treatment plants. But it could be that they were just passing through the airport, right? They may not actually be in the state. So that's always a tricky part with wastewater surveillance. But you also have people like they may work in Raleigh, right, but live in Winston-Salem. So they could be depositing in both places, right, depending upon when their bathroom cycle is. I will say, though, that most of our wastewater treatment plant sites are in urban areas. We do have some that are in more rural communities, right? So the ones that are located, for example, in the central portion of the state, I'll take it back just a second, and we can look at that because I got the wastewater treatment plants on this slide as well. But you can see that a lot of our wastewater treatment plants are located in the Charlotte region, Winston-Salem, right? And then the Raleigh-Durham. But these ones that are out here, right, in Buncombe, out the EBCI, right, up here in the northern part of the state, and also over in the Piedmont, right? These are actually in more rural regions, right? And so in those cases, it gets to be a little bit, that's where we can kind of separate out and look at kind of our rural versus urban. But it's the way North Carolina is set up. People who live in rural regions tend to be on septic. People who live in urban regions tend to be sewered, right? And so we're kind of limited with that. Case data we get from everywhere, right? And so I have case data from across the entire state that I could use to do this type of modeling. My point, because I work with wastewater, was to do a comparative analysis, and just to look at those cases that fall within those sewer sheds, right? And kind of bring those together. But we could do that, right? We could take those cases and look at them from a rural urban perspective and get a better understanding of that for sure, right? Um, yeah, it's a great question, right? And that's one of our biggest problems is rural versus urban. We just don't have a lot of sites where we can measure. Our academic partners trying to get out there and do some of this work, but it's tough. Then you lose the community-based part. The communities get smaller, right? People are more hesitant to contribute. Okay, Charlotte says, thanks for presentation. Congratulations on your work. Jessica says, hi, excellent presentation. Question from the Public Health Lab of East Texas. We have sadly lost $3 million of CDC funding to continue our work, which includes wastewater surveillance. Do you have any insights or advice on wastewater surveillance sustainability during this challenging time? Yeah, so we're lucky that we are a COE, and so we have funding, and at least for now, we have funding through 2027. But we understand that the recent cuts in public health have impacted a number of people, across the United States. One thing that I would suggest is if you were doing that sampling, at the state level, and the state lab, your state lab for public health was doing the measurements, I would really recommend reaching out to the CDC to see if you could move those wastewater treatment plants onto the Verily contract, right? They are still accepting new wastewater treatment plants, and that might be a way to continue. I understand that a lot of wastewater treatment plants are very, very hesitant to sample with a commercial contractor, even if it is through the CDC, partly because of the arrangements, the contracts, the agreements, what happens with the samples once, because all those samples are archived. So we've had some trouble onboarding people into that program as well, but we've had some great experiences with that program too. And that might be the way that you can do this. And so what I would suggest is please reach out to me, and I'm happy to connect with you and we can talk about that. I'd be happy to, to help walk you through that process, since I'm currently trying to do that with some of our sites across the state that we're looking to expand into. But I feel for you. Okay, Sarah says, thank you so much for sharing your research. Very thought-provoking. I wanted to ask how you anticipate spatial analysis of wastewater and case models might inform vaccine efficacy studies. Anecdotally, I've observed that vaccine skeptics tend not to test themselves for COVID as frequently. If this is true on a population level, it would seem to make studies of vaccine efficacy prone to reporting bias. Do you think having a robust wastewater research can improve the accuracy of vaccination efficacy studies? I think so. I mean, this is very complicated, right? We would probably need almost another meeting for this. And again, I'm happy to anybody who wants to reach out to me, please, you can nicole.snyder@dhhs.nc.gov. Yeah, I mean, this is why wastewater is great, right? Because you kind of catch everything. The problem is you kind of catch everything, right? Sometimes you catch things, then you have to deal with them. Like our recent M.P.H detection. We had a clade 1 detection that made the news a few weeks ago. And what do you do with that? And what's the public health action? But your question is great, because it is really a way that we can take what we've learned about wastewater data and utilize that to better understand what's happening at the population level. It's really interesting because for some pathogens, you can test for the wild type, and you can also do some testing for vaccine derived, right? Which is kind of interesting. So you know who's maybe had a vaccine or who has been vaccinated for something. So that's really interesting. We don't do any of that work right now, but I can see the utility for using wastewater for that, for sure. Yeah, population level is easier to convince people because it's not individual, right? You can collect that data at the population level, so they're a little less hesitant. I will say that since the public health emergency ended and since funds are being retracted, and you know, particularly for some of our wastewater treatment plants, they're you know, not willing to sample anymore. So we are losing some. We've lost a couple in the last 2 weeks. So I think that's going to be our challenge moving forward, is keeping the program going.
Great. Thank you, Nicole. We're at the hour, so great questions, great presentation. I think you really sparked some interest in our attendees. So I just want to thank you again for attending and providing us this great presentation, and wishing you all the best in all the degrees and things you're going to be seeking out in the near future. And please stay in touch with us. We always like to hear from our alumni and how they're doing.
Yeah, anytime. Always happy to connect. And thank you for inspiring me to do this work. I appreciate it. Yep. Yeah.
Thank you. Morgan,
Right.
do you want to send us off to everybody? Or?
Yeah, thank you again, Nicole, for giving us such a great presentation. I will make sure that I email the link out to everyone so everyone will be able to review the recording as well. So have a good day, everyone.
Thank you.
Thank you. Yep.
Thanks, Nicole.